our sixth presentation of this virtual annual meeting. If you are interested, if you have a question to ask, please, if you scroll down on your screen, you'll see a pop-up menu with icons on it. If you click on the participants button, you'll see a new screen pop up, and that has your name, and also at the bottom right-hand side of the screen, you can see raise hand. That's a way to signal you have a question. Or right next to that green box that says share screen, there's a chat box you can open up and ask that question, and I can relay it for you. And that's worked well also with other participants don't have very strong bandwidth to get through a video or an audio question. So Tesfaye, with that, I'm going to turn the screen over to you for your presentation on genetic improvement of sorghum for resistance to fungal pathogens. So Tesfaye Mengiste.
Okay, thanks team, and thanks for everyone who got interested and wanted to listen. I'll try to make this clear and brief without going too much into details, but sometimes maybe that's unavoidable. So this is phase 2 of the same project. We're essentially building on what we established in phase 1. For those who have not heard me talk, we focus on 2 fungal diseases. One is anthracnose and the second is grain mold. These are important wherever sorghum is growing, but especially in areas similar to the western half of Ethiopia, but wherever sorghum is growing is really, these are 2 significant diseases. And what we are trying to achieve is finding genetic solutions to these problems. So in addition to Purdue, of course, where I'm located, I think the other main drivers are EIAR from Ethiopia, Ethiopian Agricultural Research Institute, the research centers under the EIAR, Malkasa Research Center, Asosa, and Jimma. And Bako, obviously under the Oromia Agricultural Institute, Research Institute. So this is both national and the federal research institutions. And RMI University was part of this at the beginning, still part of this project, either by training students or running some field trials at a lower level. Collaborators are listed here. These are people in Ethiopia in these 3 different institutions who are helping run this project. Some of the major components of the project that involve the field trials at the different sites. And Dr. Getachew Ayana coordinates the activities in Ethiopia in collaboration with staff at Malkasa and other places, Jimma, Bako, and Asosa. And so basically, SMIL2 very broadly, our part of SMIL2 is, without going into specifics, trying to contribute to the development of disease resistance and improved materials, especially contributing the disease resistance traits to these pathogens I already mentioned. So we are, either contributing genes or materials. So in so doing, so we are helping that effort. The second part is continue to find genomic regions, genes, and develop populations. And some of these materials have been identified in SMIL1, so we try to finish those SMIL in SMIL2 in order to make them really useful or utilizable in a way. And some of you who've been in part in SMIL1 also know that when this project was initiated, Tesfaye Teso, Gabisa Ijeta, and myself, the original idea being from Gabisa, we made a large collection of Ethiopian germplasm that were characterized. Around 2,000 of those, we generated some genotypic data. We published some work on those. And we are not abandoning that, so we actually making a lot of effort to further drill deep into those materials and be able to maybe prepare them for use, whoever wants to do crop improvement work or plant breeding and genetics work. We also train people in plant pathology or genetics of disease resistance. I hope these are some of the lasting impacts of this SMIL2 project as well. So this is again a continuation. I think some of these training efforts were initiated or started in SMIL1, and we are continuing to do this as part of the second phase. These are the four specific aims, and this is just repetition of what I just described in the preceding slide in a different form. Helping the varietal development effort at the national and regional research centers, identify anthracnose resistance genes that were defined by materials identified in SMIL1. And this includes resistance genes we've mapped before and the graduate students are trying to complete now. And number 3 is to identify and characterize loci or genes that are important for grain mold resistance. And we have very disease-tolerant materials, both for leaf diseases and for grain mold, which we identified in field trials in phase 1. So we are trying to study those and figure out how to use them, whether that should be, in the form of entering a breeding pipeline, contributing the disease resistance trait again. Number 4 is going to focus on a subset of the core collection, on the subset of the landraces. Gazahing Tesema, who is currently a postdoc in my lab, have used the genotypic data generated from the larger core collection and defined 11 distinct genetic or genomic groups. And based on those information, we've reconstituted 400 sow lines, and we are going to study those in the next 3-4 years in detail. And these materials are going to be the sources of not only disease resistance, but all other traits, because these materials are supposed to capture the Ethiopian germplasm at large. So you can interrupt me anytime you have questions, if I skip over things or things are not clear. As I go along, I think they become more technical. Feel free to interrupt me, because this is online. I have no way whether I'm making a good job or not. So the first specific aim is largely based, run out of Ethiopia, coordinated from here. It's done at the EIR centers, and also Bako, which is a Oromia Regional Center. Basically, the collection of materials being tested through variety trials, national variety trials, regional variety trials, and nurseries. We started this at the beginning, so we continue to do this. The effort is focused at identifying promising materials that could either be released or at least be source of disease resistance or other traits. And these grain mold and anthracnose resistance materials have been crossed at Bako and at the national at Melkasa. And I think some of them are at the various filial generations, F3, F4, and people there continue to select and advance them in their breeding routine. As they do that, we also continue to make crosses. For sure, at Bako, they continue to make crosses to some of these materials with idea of reinforcing their varieties with new resistance genes that have been identified in this project. So ultimately, we are supporting the regional or the national breeding program, at least in the disease resistance area. By the end of this, or during this, we are identifying many loci, genes. We will be able to develop markers, and if there is the capacity there, I think people could use those materials. So this is some of the activities that Dr. Getachew sent me. Is not complete. The variety trials, amplification of seeds for nurseries. This the first season of phase 2, we're trying to amplify seeds in preparation for this upcoming season 2020, as we also continue some of our nurseries. For example, here, you will notice there were some nurseries that are over 180 materials that were selected by Habte from his GWAS experiments that were conducted at Bako and Jima. And we're going to select a subset from those, advance them, as we also maybe continue to carry nursery with a beefed up population with materials that are going to be assembled from these different sources for the next cropping season. So we've continued to do some of those trials and nurseries, but also develop materials that will enter the nurseries in the next cropping season, which is 2020. And there is more that I could not report right now, partly because the Bako, I had difficulties communicating with people from Bako. It appears their internet or phone connection is interrupted. So I will try to make an effort for those. So that's kind of a highlight of objective 1 without going in detail, but we continue to look through the data as we approach the season for the next cropping season, and select materials and advance them. Objective number 2 is identification of resistance genes, and these have 2 components. One component is basically identifying genes that underlie resistant germplasm that was studied in phase 1. These are projects of graduate student Demeka and Habte, plus a postdoc in my lab, Gazain Tasamma. So we are almost at completing those projects. I will describe them a little bit further. So that's part of this objective 2. The second part of this objective 2 is we have new materials, and those materials were identified based on nurseries run in Ethiopia to be found to be very resistant to both leaf and grain diseases. And we are now developing populations in order to be able to identify those genes and then eventually prepare them for potential utilization. I don't want to go in detail, but I think these are the 3 specific things we are trying to do here. As I just said, we either are close to finishing some of these projects or are just starting generating a mapping of population. So the ones that are being finished are were conducted, are still conducted by grad students. The ones that are being initiated are conducted by a postdoc here, Gazahin, and they will be sent for further characterization. Part of them at least will be sent to Ethiopia to be followed by newly recruited graduate students, and part of them will be studied here at Purdue. If you remember me talk before, for some of you at least, in phase 1, we identified one important anthracnose resistance gene. It is the very first one we identified, so we called it ARG1. And we've finished that work. We still have a low-level experiment going on to complete the story, but that's a done deal. Demeka, who is a graduate student, as I mentioned, works on 2 resistance genes, called them ARG2 and ARG3. And he is on track to finish his thesis this summer. Hopefully, if it was not for the current issues, maybe he would have been able to do it this semester. But he's on track to do that, and following him, I think the work on this will be completed. He is on the validation stage. A 4th gene was identified by Habte and the Gazahin. I think initially this work was done by Gazahin, and the Habte took it up. So he's done the genetic mapping, the validation is underway, and hopefully this will be also done by the end of the year. So in addition to this, we are generating new mapping populations based on lines that show grain mold and anthracnose resistance, which is dual resistance, and those materials will hopefully yield at least 4 other genes. So that's, I think, eventually a very good work, and I think potentially useful, whether it is in Ethiopia or anywhere else where these diseases are actually challenging. So, and this is, I think, familiar maybe for some of you, the ARG1. I just wanted to briefly remind you, this is a very resistant material. It's actually a field trial in Bako, and the resistance gene was cloned based on this material. So this is an excellent source of broad spectrum resistance. And if you look at the panel here, you see that actually a lot of materials that carry this resistance gene actually are very resistant, where are multiple materials that do not have that resistance gene, or the gene in these lines are defective, actually very susceptible to these materials. So this work has been validated, and the people have actually started to, a long time ago in Ethiopia, started to cross and entering the breeding program. This is work from Demeka on ARG2, and you will see that here, this is a germ, a line that carries a resistance gene, and this is a sibling that doesn't carry the resistance gene. And you see that the differences are significant, whereas this material is able to restrict fungal growth, and the susceptible material is overtaken by the disease. This is a clear difference in disease symptom. And what the Demeka did was then stain these materials to see how much fungus grows in the tissue, in the infected tissue. Here and here, you'll see that this fluorescence substance basically corresponds to the fungal growth in the susceptible material, whereas here, the limited fluorescence is a mark that this resistant line actually is able to contain the fungus and is able to resistance. And these 2 lines actually differ not by a lot, they differ by a single resistance gene based on the Demeka's recombinant inbred near isogenic lines that he generated by crossing. Just to give you further example, another resistance locus, ARG4, a collaborative work between Habte and the Gazain, and you'll see here that in the susceptible background, you see a larger disease lesion where a fungus actually sporulates, whereas in the resistant material, the material is able to resist the fungus. So this is just to highlight some of the strengths of these materials. And this again is a slide from the Mecca. You'll see that these are two lines that only vary by a single gene, because these are near isogenic lines, close to a single gene, I would say. And you'll see that when the Mecca stains them, the susceptible, the guy that does not have the resistance gene, allows the growth of this fungal structure, which is typical of the anthracnose fungus on sorghum. Whereas its sibling actually that is resistant, that carries the ARG2, is completely immune and it doesn't let the fungal growth. This is just to highlight the strengths of these materials when encountered with the fungus. And in nature, we actually challenge these materials with a lot of fungal spores, which the plant in nature would never encounter. And we test these materials under conditions that promote disease. So we really challenge them to see the effectiveness of these resistance genes. And one of these, again, the ARG2, and shows that, you see, not only is the leaf resistant, but also the stem, the stalk is also resistant. As you can see here, the susceptible plant actually shows this disease symptom on the stalks as well. So this is remarkable resistance level. So the Mecca went on, identified the gene. So this is too much for a single slide, but the point is he mapped it to chromosome 5 and then zoomed into the region of chromosome 5. It's like finding, you know, a needle in a haystack. That's what it amounts to, and it's a relatively time-consuming process. Once he maps it to a certain area and then using genetic approaches, he zoomed it down to, you know, multiple, many kilobases, maybe hundreds probably, and eventually he could see the difference between those susceptible and the resistance lines is basically due to polymorphism or difference in this gene labeled here, NLR, which by traditional plant pathology is called the resistance protein. So eventually the Mecca was able to identify the specific resistance gene that makes the difference between those materials. And this is just to give you an example, so this is the same kind of approach for cloning the ARG4. We did this before for ARG1, and the Mecca recently completed work through a similar approach on a third resistance gene called anthracnose resistance gene 3. So I don't want to go through this, but this will give you the flavor of the things that are going on with graduate students' work here at Purdue. So what does all this mean in terms of utility? I think we will, by end of SMIL 2, we hope to have identified and characterized many independent genes, and that can be used. I think potentially, I hope a lot of, we'll try to promote the utility of these genes in Ethiopia or other ways, other places. When the publications come, it will become widely distributed. Hopefully it will be easier. But I think we've made significant progress. If you look at the literature in wheat rust pathology and the stuff, it took many, many years to define, obviously hundreds of genes, but we've started recently, with the SMIL 1, and we have never done this before. So I think we made a lot of progress, and right now we are very, very confident we'll be able to finish those and publish them. And I think we'll open the way for more research, because although a lot of people study pathology, anthracnose pathology, also resistance, beyond sort of characterizing germplasm, I don't think there is a thorough, work in terms of defining genomic regions and to the gene level comparable to what we've done here. So that's encouraging for us. So the third objective is on grain mold resistance, and some of these materials are also resistant to anthracnose, and we are generating recombinant inbred lines to identify grain mold resistance genes in addition to anthracnose. And some of you know grain mold is not the easiest beast. I think it's a little bit challenging because the resistance involves many genes, but I think these materials are very good for pursuing these kinds of objectives. So 4 different objectives here, and the one is basically something that's done in Ethiopia, being done to try to find mold-resistant white and yellow-seeded materials. The second one is basically identify genes for grain mold resistance, and through recombinant inbred lines or biparental sequencing. And the fourth is what Habte has been doing and is currently trying to wrap. Among the core collection, he's selected sort of 700 or so that are relatively early to medium maturing, and he has conducted GWAS to identify loci. So that part of activity is also part of this. That activity is also part of this objective. So in the nurseries that were conducted at Bako and the Jimma, some of these materials were very, very clean. And this is a picture I took myself when I visited Jimma 2 years ago, I think. And you will notice that some of these materials are very clean. They show resistance in the leaf as well as in the head. And these materials originally came from the Bisas breeding populations or collections, and really high level of resistance. So these were tested at Bako and Jimma, and because they were interesting, we wanted to pursue those because maybe these materials are not good enough to be released as varieties. These are not improved varieties. These are, I think, landraces, some unimproved materials collected from somewhere. So we wanted to know why these guys are resistant so we can use them in breeding. So these were the lines. There are multiple of those. You see that there are anthracnose resistance, there are grain mold resistance in the grain. Some of them are actually broad spectrum resistance. So we wanted to pursue this. Here, what we've done is we've started to generate a mapping population. So they were crossed to another line, which is typically susceptible and also shows good level of polymorphism with these resistant lines. So the idea is two. One is to develop recombinant inbred lines, and we can send those to Ethiopia and they can be used. The other is from those crosses, we will use the F2s and the progenies of the F2s, F2-3s for running what's called the RNA-seq to identify why are these guys actually resistant to anthracnose. But the mold resistance gene cloning will be based on trials or experiments conducted in Ethiopia largely, because that's a complicated assay to do it at Purdue. And also it can be done here, but I think the fungal strains will be different. So the relevance may be really limited. So hopefully by 2021 cropping season, we are hoping that some of these will be at F6 or at least F5 level so that they can be pursued in Ethiopia. We are doing here in the greenhouse, try to squeeze these plants to give us seeds at least 3 times or so per year in the greenhouse. So we are hoping to be able to do that. So these materials will be good sources of resistance, and if we have the recombinant inbred lines, I think we have multiple lines with different resistances that could be used not only for gene identification, but also for a crossing program. So this is a repetition of what I just said. This is a slide from Gazain, who already made crosses between these resistant lines. So at least 4 different populations, mapping populations, are being generated. At least we will deal with 2 of these here, but 2 of these will be, I think, studied by hopefully potential students in Ethiopia. But all the recombinant inbred populations that will be generated in this effort will eventually end up in our tests in Ethiopia in the western regions. So in parallel, as part of this mold resistance studies, Habte took a subset of the large landrace collection and planted them at Baku and Jimma, and he had 2-year data and did mold resistance at different times in the field, plus in the lab, plus on the panicle after and on the grain, and did some association mapping. What he showed here is just as a confirmation, or at least the thing, our thing is working, our approach is working, is that one of the things he identified is that the polymorphism in the TAN gene, TAN1 here, actually correlates with resistance, and this is not surprising because other people have actually demonstrated that. So it suggests that by doing a similar work in the field, you can identify loci that are important for grain mold resistance. And these are 2 other loci. One of the polymorphism identified is actually closely linked or in the ABC transporter. And these are also known in the flavonoid and anthocyanin biosynthesis pathway. That again, these are molecules that have been implicated with grain mold resistance before, or at least fungal resistance. So it sort of gives us confirmation that we are on the right track. One of the things he also did was, if he selected a subset of the materials and did it at GWAS, and he could identify different kinds of genes, in this case cytochrome P450. Some of these kinds of genes have been implicated in Fusarium head blight, in I think on dawn resistance. I mean, this is a pathogen that is very, very similar to sorghum grain mold. This happens, this is caused by Fusarium in barley and other small grains. So that again suggests that what we are doing is on the right track. We can identify, using association genetics, we can identify loci that could be used, and eventually that those loci could be helpful for designing markers if the breeders are actually keen to pick up some of these materials and some of these, at least the very clear ones. So just to highlight some. Habte also did a little bit of work towards understanding what goes on during grain mold. Grain mold, as I said, is very difficult disease. It's caused by many, many different pathogens together, a lot of necrotrophic pathogens. And we wanted to understand, I think, what are the processes, what are the kinds of genes that are induced or suppressed during grain mold development. In this case, when infection occurs as the grain forms, at the early stages of grain development, what Habte did here in West Lafayette is he planted two different lines, known lines. One is called RTX430, a known grain mold susceptible material, and RTX2911, a known grain mold resistant material. And he infected those with a soup of combination of different fusarium and other fungi, and inoculated the panicles and harvested the developing grain, extracted RNA, and then compared the global changes in gene expression. And you will see that there is a clear difference in terms of gene expression between the susceptible material and the resistance material. And he has a lot of cool figures he generated to understand what perturbations, what kinds of changes happen that correlate with resistance or susceptibility. And that would really shed light on the physiological events or cellular events that occur when a developing grain is infected with the fungus. I don't think people have studied this because these are challenging diseases. Grain is a maturing tissue, so it's very, very difficult. So this is sort of kind of basic work that will illuminate what's happening. Can we identify genes by this mechanism? And indeed, he identified multiple genes that correlate with resistance, that are highly activated in the resistant, but not in the susceptible. And two of these are highlighted here as an example. These are what are called the defensing genes. You will see that the black bar is actually the resistant material 2911, and the white bar stands for the susceptible one. So in the resistant material, you'll see that this gene, which is known to be killing fungi, is actually highly activated. And this is another defensing gene. You see that in the resistant material, actually, it's highly induced, whereas in the susceptible material, and it is not induced. I'm not saying that this explains the clear difference between these two lines, but at least these are some of the things that may contribute. The resistant line may need to activate a lot more genes. So it is not one gene, just like the anthracnose, that's required for resistance. Multiple genes. Genes like these contribute and then orchestrate resistance to this mold fungi. So this work by Habte and is part of his thesis, and I think he's currently trying to finish using this occasion where we are not on campus, trying to write a paper and finalize a story publication towards his thesis. So implications of this work is not very different from what I said before. Basically, we are generating materials, identify germplasm, lines, develop recombinant inbred lines, identify genes or loci, identify genes through RNA-seq, and all this will hopefully in the medium to long term will facilitate the breeding effort. At least that's the intended goal in the long run. So this is aim 1 through 3 so far, and aim 4, as I sort of highlighted before, focuses on 400 lines that were selected from the 2000. And this will be, the hope is, these materials will be the foundation of the genome, the foundation of breeding effort, whatever approach you take. These are rich, these are sources of various traits, not only disease resistance, they represent everything. And the 400 lines where we think will capture the Ethiopian sorghum germplasm, more or less. That's the idea, at least, okay? So this season, the main activity on those was planting and seed amplification, so that we have seed enough for multi-location tests. So that was done at Malkasa. So we were able to harvest seed based on a report I just received from Dr. Getachew, and also Habte's communication. 300 lines we could harvest. About 100 lines failed, mainly because of issues related to cold storage at Malkasa. I think their cold storage failed. That's where these materials were stored. So what we are trying to do now is not the greatest news for us. Try to recover the 100 lines from the previous harvests, if possible, at some point, as we advance these 300 lines forward, starting this cropping season. So these 400 lines will be really studied very closely in much detail, plus also a subset of these, at least half of those will be, will be generating whole genome sequences. We have a GBS for most of these lines, but the GBS is not sufficient. I think will be good to generate whole genome sequences that will be useful for a lot of people, I think, who are interested to work on these materials. So previously, just to remind you, we had around 2,000 lines picked from among 9,000 that's kept at the Germplasm Center at the Gene Bank in Ethiopia. So the 2,000 were characterized for various traits and showed very high diversity, all sorts of traits from flowering, maturity, plant type, anything you take, there is a lot of diversity. I mean, anything that we could measure. I think there are a lot of things we didn't measure. We think there are a lot of, this material is rich in a lot of traits. So we've characterized this. We've sequenced 1,600 lines GBS, and that's the basis for doing a lot of the GWAS. And this work was published last year, I think, and in collaboration with our collaborators in Ethiopia. That was good. One of the things that during analysis, Gazain, who did most of the analysis of this work, using the GBS data, is that a lot of the varieties that were released in Ethiopia are actually not very diverse in terms of their backbones. As you can see, I'm not sure you can see that. Something is preventing me from, let me see, yeah. That you see that if you look at sort of the blues and the grays, if you consider these are the genomic backbone or the bloodline, a lot of the materials that are released in Ethiopia, these are the names of the cultivars. I think they appear to be similar. So I think so that's a narrow genetic base. So one of the things that this work hopefully will impact is inform people that this is what's occurring, and actually the current collection will boost the diversity of this material. So this could be one way of enhancing the variation in the breeding material. It's to use the germplasm here. So the other thing Gazain was able to do during, and this is not published, I think, is compare the SMIL collection with the collection that is somewhere else, including the Sorghum Association panel and the germplasm that is held at USDA and the National Plant Genetic Center. So there are a lot of sorghum lines that are held by USDA that came here a long time ago. So when he compared these three populations, obviously what he observed was that the collection held by USDA and the SMIL collection actually are very, very similar. They cluster together, whereas the one by this SAP, the Sorghum Association panel, appears to be distinct. So for people who have a hard time working on Ethiopian germplasm, I think this is the place to go. They are very similar to the SMIL collection, except that this is currently in Ethiopia and this is currently in the US, which is more accessible. So this is very, very interesting information. So this work was published on, I think we used that collection to identify some mold resistance. That was a work of Habitat that was published. And also the large scale collection was published we mapped a lot of genes, not only not disease resistance actually, but other traits. So that's out in the public sphere, so people could be informed. And more recently, Gazain, what he started to do is, since we have the information, the point of origin of these materials, he was doing association genetics, using the altitude, temperature, other variables, climatic variables from where they were collected, and they try to see if if these materials actually have different adaptive traits. So let's say some are collected from high altitude area or low altitude area. What makes them different in terms of adaptive traits? And this is a kind of GWAS he did. This is not really published yet, but we are working on this. Indeed, he sees some genes that correlate with altitude or with temperature. I think most of these are, he tells me, are genes for abiotic stress tolerance. So it makes quite a lot of sense. I think these materials coming from different regions would have different genes that would confer different adaptive responses, different adaptation abilities, really. So we'd like to pursue this line of research a little bit, if possible. So the core and the GBS and what's the future here, I already, I think, highlighted it a little bit. Basically, we have to be very careful in further characterizing this in more detail, and to be able to catalog this, link the phenotype and the genotype. And genotype here will be whole genome sequencing of at least 200 lines, and anybody could access them for utilization purposes. So that's what we plan to do. This will be done at Bako, Jimma, and Asosa. And I see some students are interested from Ethiopia, so they could really use it for other traits as well. We will be measuring disease resistance trait, but also a lot of other traits, including morphological traits, yield and yield components. So we will be doing that starting this season, with the caveat that they may not be complete because of the seed shortage I just mentioned with the 2020. But in the next 2-3 years, I think they will be completed. And this will be replicated trials at 3 different locations. So it's a lot of work actually, but we have recruited students to help us with this effort, and I will explain that further. I don't want to go through this because basically these 400 lines are able to capture, we think, the Ethiopian sorghum germplasm. So that's the logic behind, you cannot use the 9,000 lines that are in the store in the germplasm center. You cannot even use the 2,000 lines we collected. That's too cumbersome. So what we are trying to do is distill it down to essential components of 400 lines that capture the whole collection. And these were grouped based on their genomic. So there are 11 clusters, which means 11 kind of bloodlines that are different. So we are taking, from those 11 representatives to constitute these 400 lines. So this will be sources of everything, I think, in terms of traits. So these are sort of the technical objectives I highlighted. I think this year will be critical for us, as potentially we'll have a lot of results to publish and students to finish. And what else we did? Basically, we did training, promised training in SMIL phase 2. So we promised 2 to 3 training workshops. I will tell you that. We also promised 2 new PhDs in local universities. We'll tell you that. And also promised to complete the students, to graduate students that started in SMEAL phase 1. So in August, the group here traveled to Ethiopia, and we offered a workshop on genomic approaches in crop improvement. That was a very exciting event, I think. A lot of young people were recruited from EIR, Oromia Research Institutes, universities, Institute of Biotechnology. I think people from here, we all traveled and gave training on, basic bioinformatics, molecular approaches, various topics, and also invited people from Ethiopia, well-established researchers in sorghum pathology, breeding, genetics, weed science, and these people from various universities and EIR gave lectures. So the young people were very excited. I think this was very popular. The feedback was good. So I think during the second phase, we promised 2 or 3. So we're hoping to do one on pathology either this year or the following year, depending on the situation going on now. I think we would like to give one on plant pathology. I'm not sure it will be a large number like this one, but hopefully during the cropping season, but if not, we will see how. But we will plan that. I think coming summer, we will decide whether that will be offered this year or the following year. So that's something we did. Graduate student, we also promised to train 2 students, PhD students at local universities. So those positions were announced through EIR and Oromia Agricultural Research Institute. So 2 PhD candidates were selected. Chameda Brhanu joined Haramaya University, and Asafa Gidisa from Asosa EIAR joined AMBO, and these are paid through the SMEAL, supported through the SMEAL funds. And there are 2 other students who were part of this SMEAL project. One of them, Mogas, he joined Addis Ababa University. Another student, Kabadir Saleh, was also very key person at Bako. He joined University of Nigeria. These 2 students are interested to conduct research with us using some of the materials we generated. And I'm happy. I think the project will benefit if they actually be involved, whether it's disease resistance or other traits on this collection. So we are trying to promote that support, at least the research, helping them, giving them materials and providing some ideas. So people here, Habte, the Mecca, and the Gazany are also chipping in, trying to help these people, either sort of developing the materials or just giving them ideas, sharing them ideas they learned here. So we're trying to promote that, because these materials, the core and the subset, there is some level of genotypic data or information. So there are a lot of students that are getting in touch with us, trying to use the material, measure, conduct their assays, and then use our data to establish, the kind of work we were already able to generate. So that's actually good. So whoever is interested, I think we'll be able to support. The only limitation will be the seed. I think we will work on that during this summer and during the upcoming summer and the following off season, I think, in Ethiopia. To complete graduate students, I think 2 students that were started in SMEAL 2. SMEAL 1, Habte will finish in fall. The Mega indicated will finish, I think, summer 2020. And I think that's the promise that was made. That's what's going to happen. In terms of the next season, it will be I think I mentioned most of this, a continuation of what we've started or beefing up a little bit. We have a lot of field trials being planned, nurseries, multi-location and replicated trial of the core, and completing the recombinant inbred lines and send them to Ethiopia. Completing work on ARG2 through ARG4 here by graduate students, and potentially holding a plant pathology training workshop, and also, at least one of the PhD students will be offered a short-term training here. If they are able to come up with their materials, I think they would be able to come here for several months and work on something they cannot do at home. So we'll try to facilitate those kinds of work, plus I think more. Challenges, I think challenges are there, not only the current situation, but persistent challenges, staff turnover, I think that's always there. And people move on, people go to trainings. And during that, I think follow-ups, smooth transfer of data and the materials is an issue. There is not much we can do sometimes, I think. Issues with cold storage of seeds, I think that's not a good thing for the last time this happened. I think they have solved this problem, but that is a hiccup on us. Challenges with import of seeds, fungal strains, tissues, that's always a problem. I think there are things we can do here that they cannot, our collaborators in Ethiopia cannot do, but there is always an issue in terms of MTAs, complicated stuff. Even let alone seeds, fungal strains, we have broad fungal strains. Through permission of the USDA EFIS, we have the permission to do that, but the system there does not allow us to bring a lot of fungal strains, and the MTA is complicated, so that's an issue, I think. So I think those are the main challenges we have, and hopefully we'll overcome some of those. And I think I would like to acknowledge SMIL and the USAID for the support, Purdue, and I think EIR, BACO, and all the people that are involved in this project. I think I have not covered everything, but I think I touched upon things. So I will stop here and let you ask your questions if you have one.
Well, thank you, Tesfaye. That's a very good and comprehensive presentation. We've got some questions that have already been submitted through the chat box, and so we'll go through those first. Early on in your presentation, and Jito mentioned, submitted a question. In it, he says that you identified leaf disease genes in SMIL. One, did you identify leaf blight resistant genes? And this comes from a question that came up yesterday when the sorghum pathology project in West Africa was presented.
Yeah. So, the ones we test here for leaf disease resistance are specifically anthracnose, because those are the fungal strains we test. But when we conduct this research in Ethiopia, and some of them are very, very resistant, I say they are resistant to foliar leaf diseases. So that's because they get all diseases that are out in the field. So not only just anthracnose, but other diseases. So that's why I made that distinction, leaf diseases. So anthracnose is one blight, but there are other diseases. Rust is another one. Bacterial blight is another one. Some of the lines that were tested at Bako and the Jimma are generally resistant to leaf blights. The ones we identified here are specifically resistant to anthracnose because that's what we tested. And among those, like ARG1, are resistant to rust plus other diseases, leaf diseases as well. But ARGs 2 through 4, they were specifically tested only for anthracnose resistance. Does that answer the question?
Do you know any follow-up clarifying questions?
It's okay for me.
Okay, thank you. Then there's a couple of questions on anthracnose specifically. one by Peter, and a complementary one by Bettina. Peter writes, is there significant genetic variation in the anthracnose that infects sorghum in Ethiopia and globally? If there are many anthracnose biotypes, what does this mean for achieving durable resistance in Ethiopia using the resistance genes that you've identified, as well as in wider geographies?
Yeah, so yes, I think that's a fair question because there's a big variation here. So the way we address this is we have strains that are collected from Ethiopia. They are not enough. We test some of our materials with those. We are hoping to get more. So whatever is identified here will be tested for anthracnose strains from the US plus some from Ethiopia. But eventually, I think it's good that some of these materials have been tested in Ethiopia. They are resistant to Ethiopian strains in the field. So they are fine, so they will be useful there. Some of them were not tested, and that would be the next step. But some of them, luckily, like ARG1 is broad spectrum resistance as far as we know, we tested. So that is a fair question, and we try to address that specifically because eventually the project target area is Ethiopia, so it has to be useful. And that's how we try to address is basically using Ethiopian strains or field tests in Ethiopia.
Good, thank you. I'm gonna offer the microphone to Tim Lust. I see your hand up, Tim.
So my question was similar along the lines of ARG1. And if you're just taking genes, I understand that. But if you're using that material in a cross, is it the same? Is that a waxy? Is that a waxy hybrid?
No, these resistance gene is available in different sources. And these are either in landraces, these are not in hybrids. The one I described, there are multiple of those in different materials. What I was talking is basically a gene in different backgrounds that confer resistance to leaf blight plus other diseases. So it's not in hybrids, it's available in a range of lines, sorghum lines.
Let me ask my question differently. I believe if it is the ARG1 that I am familiar with, it is a waxy line. And so just be aware that if people are putting it in a breeding program, there may be some other characteristics or some other challenges with that line. It may be really good or it may not be good, but there are some other background things if that is the line that I think it is.
I'm not sure I understand fully the question, but I don't think it has anything to do with that. But I could be mistaken but I understand this gene is available not just in one line, but in multiple different lines. We've proven that. And any of those multiple lines could be used as a source of resistance. So people could use different sources for crosses. The only thing they carried that ARG1 resistance gene. So this gene may be available in some good cultivars, but also cultivars that are not really good by other measures, by agronomic traits. But the only useful thing from these materials I indicated are the ARG1 disease resistance gene, which is a classical disease resistance gene. So I do not think they are related to the waxy trait or any other seed trait.
Thank you.
Thank you.
Great question. Other questions for Tesfaye and his team?
Yes.
Any hands going up or any questions written in? Bettina? Bettina has 3 questions for you. Bettina, I'm going to let you go ahead and open up your mic and ask these then.
Yes, Mengiste, many thanks for this really impressive work. So one question is, are there easy to use tools for African sorghum breeders to employ the resistance genes you identified?
That's a really great question. I think we know this is something that I always struggle with. And, I think the anthracnose is easily tractable because it's, you could find a single locus, single gene that really confers resistance. One is ARG1. So they can cross it, either select by markers or follow the trait through field-based screen. So those kinds of easy. The challenge is a grain mold because I don't think one gene is enough. So you need multiple genes. How do you select that? So they can still do that, but I don't think they can maximize what we've found. So in the long run, the solution for that is, I think, to build up the capacity to improve the local capability. And these are not difficult things to do, but I think there must be a facility, and there are biotechnology labs, but they are not collaborating with plant breeders because the labs are somewhere else, breeders are somewhere else. But in the long run, I hope this will be used. The anthracnose, I have more confidence because I think these are controlled by major genes. They are not difficult, whether you do it by classical breeding or use markers.
Okay, yeah, I understand.
Yeah.
The other question, can you explain to me like the role of tannins in the resistance against anthracnose and grain mold? Because what I observe, it's really fancy to select the low tannin sorghum, but would that have also implications for resistance, or how do you see this?
Yeah,
I'm not really an expert in this.
No, I'm also not a good expert on the biochemistry of those things, but there are obviously data that show, tannins protect against fungi, and there's a recent paper that also demonstrated they also protect birds. So they are there for the plant has developed them to fight pests, whether they are animals or fungi. But the bad thing is also, I think some, they are associated with some anti-nutritional properties. So if you try to breed in tannin, then you lose that nutritional quality maybe. I don't know how universal that is. So clearly they are linked, but in Ethiopia, if you look at, what are best for injera and other things are not, with tannin. So people like the white ones or the yellow ones, and they have I think in some places they prefer those, but those may be a little bit mold susceptible. So what we are trying to do in our case is, in addition to these kinds of things we found, screen for white or yellow seeded materials that may or may not have tannins, and then find those white seeded, yellow seeded that are maybe more resistant. I know it is challenging, but we are trying to do it, to take a genetic approach. So tannins confer resistance, but they also have other issues with them. And if you notice one of the pictures I show, a very nice material that is very resistant to, to grain mold and anthrax, has, brown seeds, a lot of those, may have tannins, but there are also those red or brown seeded ones that don't have tannins. So we have classified those. So these are unfortunately complex issues. You try to improve one, you affect the other trait. So we have to.
Yes.
But there are, places that, tannins are also okay, I think.
Yeah, but for if we adopt the market-oriented approach, I think we need to sort this out, right?
Yes, I'm not saying that. Yeah.
Low tan resistant materials, if that's possible at all. That was mainly my question. The other question I had: do Ethiopian farmers appreciate the resistant sorghum materials? Or
Yeah.
are there any other trade-offs that need to be considered before those resistant materials can really be adopted?
Yeah, I think so. I mean, I think not everywhere, but this project started off focusing on the western half of the country, where the limitations are, you know, they have rainfall, they have everything else, but diseases are important. And
Yes.
those diseases, mold, you know, like areas like Pawe, Bako, Jima. I mean, this Jima is, I mean, a disease-prone area. You know, in some years you cannot grow plants, mainly because of disease. So I think diseases are important in those areas, but not everywhere. I think, I don't know who is listening here, they can comment. You know, the northern and the eastern, the northern part, you know, there are other issues other than diseases. But even in those areas, in some seasons, I have seen diseases, but there are, I think, disease issues, and the farmers recognize that. And I think the research centers work on that trait now, I think. So the answer is yes.
Okay, thank you.
You're welcome. Thank you very much.
Other questions from the audience? Peter, was your question sufficiently addressed by Tesfaye?
Yes, I think so. I mean, he recognizes that there's a, you have to take into consideration the variation in the anthracnose fungus. And
Yes.
so his results use the local fungus strains, so they apply their broader application of the results would have to be verified. If that's a proper summary, is it, Mengiste?
Yeah, no, you're right. Yeah, I think you said it. We try to verify, we've tried to use the local strains, we try to do field tests in Ethiopia.
Yeah. I guess maybe a related question is, do you understand the mechanisms of the 4 different anthracnose resistance genes? Are they different? Are they complementary? And would stacking them somehow confer greater durability, broader application?
Yeah, that's a very good question, really good question. We know that these are independent, and that's what we know. You know, some are relatively broad, some are relatively very specific, which suggests that if we cross them and generate, you know, lines that, you know, that are stacked, they would offer broader protection. So that's, I think, a good question you raised. And in the long run, I think when these things are, you know, are published and we are trying to generate some crosses, I think that's the ultimate goal, because also when we know these genes, it's easier to stack them because we have markers to track them faster and easier. It shouldn't take generations. I mean, it shouldn't take too long compared to if you do it traditionally. And sometimes you can look for those genes even at the seedling stage without planting them in the field. So that's the idea. Yes, you know, my hope is in the long run, specifically when we know the particular genes, we will be stacking them. Stacked materials will have a broader resistance than one particular gene alone.
Great, thank you so much.
Tesfaye, question for you. I guess I didn't address this in your presentation. How do you anticipate that the COVID-19 pandemic is going to affect your activities this year? Have you given that any thought?
It already affected us, right? was not a good thing that the SMIL meeting was cancelled, because that's, I think, where we were, you know, sort of discussing extensively and stuff, review things, revise things, and plan the future. It affected us, but, you know, once we go through this, maybe the first thing I would do is I think myself, or at least a couple of us would travel and try to do things we couldn't do. The season is coming up on us. I think that's a little bit a challenge. So we will increase our communication a little bit more. I think through Malkasa, it's a lot easier to communicate. Baku has been challenging because I think the government has cut internet and phone services from the western parts of the country. That's an added challenge. So the first thing I would do once things open up is travel immediately, but also, maybe increase my travel despite the responsibilities here, including teaching, adjust things a little bit differently.
Okay, a lot of unknowns, it appears.
Yes.
Okay, the floor is open for questions. If not, we'll bring this presentation to an end. Last chance.
Yeah, let me ask a quick question. Now, Hapte is working with you, right? He's fully engaged in this program. He's also engaged in the program that he's doing with Remu and Gabisa.
Yeah.
I'm wondering how functionally and operationally, how is your work feeding into the work that that team is doing, that Gabisa, Remu, and Hapte are doing? Are you helping them screen materials that they are advancing? Are you providing them assessments of some of their parental lines? How do you guys fit together? Because clearly the potential complementarity is great.
Yes, I think the core collection was an overlapping objective in the first phase. And potentially the core is not only just a disease material, but a lot of other traits that can feed into a lot of breeding effort. And Alamu mentioned the other day, and this will be really a good source of material for a lot of the breeding efforts. And Hapte is good as not only as a student here, but it connects us to the different angles because he used to coordinate the national program, so he knows a lot of stuff. So, for example, in terms of breeding and hybrid breeding program, if a hybrid breeding program is going to be targeted to the West, I think in addition to those other traits they work on, clearly they need to include disease resistance as well, because these traits are important in that region. And I think as these things come to conclusion and we publish stuff, we share material, I think we can cross them, we can interact. And a lot of these later materials, I indicated, also originate from the Bisa's lab. So it helped a lot. So we interact in terms of advancing the overall goal, although my focus really is disease resistance. But when you look at the core, it goes beyond the disease. Disease is one aspect, but we study a lot of other things. In fact, we try to recruit students who do other things, including physiological studies, potentially striga resistance and other traits. My part of SMIL cannot do everything, but that material, we do the whole genome sequencing and the stuff can be very useful for a lot of other traits. So that's, I think, how we interact, and Hapte, is in constant contact with national programs, so he facilitates that interaction.
Peter, is that you need to follow up with that, or are you good?
Absolutely. No, thanks so much.
All right, I don't see any more hands popping up on the screen. So I
have one more question.
One more, Bettina. Okay, thank you.
Yes. Do you have any linkages with the Mercy project?
No.
In sorghum breeding?
Not directly, but I talked to Alamu the other day, and so my project, they don't do the disease. I don't know the detail, but disease came into play only after a SMEAL project. So any breeding effort that again targets that area, I think will be good for them if they consider our materials. And we will be happy to help to facilitate. Although at this stage we don't have a direct interaction, but the national program is there, I think, to facilitate some of these things. I would be really excited if they pick up some of these materials. And my job is also to try to publish this and promote the use of these materials, because there was a lot of effort that went into that. So, and Alamu, the coordinator, knows this. Habte knows, and he's in constant communication. He knows this. And as these results come online, I think people will also likely be using them. But obviously, right now, I don't have a direct interaction with this Mercy.
Mm-hmm. Okay, thank you.
Bettina, I think it's very important to mention that the predecessor to the Mercy Project, Sheila, all of the work that the Australian team did to build capacity within EIR, especially the electronic data collection activities and the analysis of data, were extremely important for the broad scale phenotyping and the collection of data that took place with the core collection. So I don't remember if
I imagine
Alemu or Habte who has mentioned that several times, and I think it was fortunate timing with
Yeah.
Okay, thanks.
Thank you.
Well, thank you, everyone. It's been another very interesting and exciting presentation. I do have to say that the benefit of the virtual meeting is that we've had plenty of time to talk and discuss. Presentations have been going almost always an hour and a half or so, even longer. So we'd have never been able to do this face to face. So we've gained on this side. Tomorrow we'll have 3 more presentations. Bruce Hamaker, Mitch Teinstra, Jeff Morris, West African breeding and food processing presentation.